Alright, hello, welcome to another adventure of anatomy and physiology with Dr. H. Today we're going to be discussing the sarcomere. And the sarcomere is the smallest functional unit of muscles. Functional hopefully makes you think of physiology. And so if we're thinking of function, we should be thinking of how things work. But what do we know about muscle? What does all muscle have in common? It can contract and relax. It can produce force, and it can release that force. That's its function. Therefore, the sarcomere, if you think about it logically now, what's the function then of the sarcomere? This is the property of muscle that allows it to contract and relax. This is the property of muscle that allows it to finally create force and then release that force. So let's look at what muscle is made up of. Okay, if we think of skeletal muscles, which is what we're going to focus on here, skeletal muscles, a big skeletal muscle now is made up of multiple bundles of fascicles. Each one of these fascicles is made up of multiple muscle fibers, and muscle fiber is just a fused muscle cells, right? Multiple muscle cells, hundreds of muscle cells that fuse together during embryonic development. So this is just our fancy word of saying this is a big cell, right? It can be up to 10 centimeters long. Well, what are muscle fibers made up of? Muscle fibers are made up of lots and lots of myofibrils. Now these are tiny little packages that we call myofibrils. What are they packages of? Inside of all the myofibrils we have multiple myofilaments. These are the proteins that drive the function of muscle. Okay, it's the myofilaments that do the function. So that's why you guys need to know about them. That's why you guys need to be able to draw them. So let's take a look at the myofibril. And if you actually look at an individual myofibril, what you're going to see is a ziggy-zaggy line, and a ziggy-zaggy line, and a ziggy-zaggy line, and there's multiple repeated Z lines over here. These are called Z discs. Okay, the ziggy-zaggy lines are called Z discs. And in between the Z disc, you have a dark band, a dark band, a dark band. And surrounding the Z disc, you have a light band. So this is what we're going to call light, dark, light, dark, light, dark, light, dark. And if you look at muscle underneath a microscope, it's one of the most organized structures in nature. Light, dark, light, dark, light, dark, light, dark, all the way down this myofibril. The light portion is called an I band. Okay, this light portion right here, this is called an I band. And then the dark portion is called an A band. This stands for anisotropic. Any word in Latin that starts with the letter A or AN means without. Without light, with light. So A band is without light. I band means isotropic, which means in essence light can pass through it. And if light can pass through it, then it's really light in color. And if light can't pass through it, then it's going to be dark in color. So the I band and A band. So now let's look at it like light, dark, dark. I band, A band, I band, A band, so forth, so on down the row, repeating patterns. And what's separating each one of these? Z-disc. Okay, there's a Z-disc right in the middle of each I band. And actually how we define a sarcomere is from one Z-disc to the next Z-disc. The sarcomere is one Z-disc to the next Z-disc. This area under that portion of the curve, that's a sarcomere. And that's what we're going to zoom in on and try to describe a little bit better down here. So what you see here is one Z-disc and the next Z-disc. This is Z-disc to Z-disc. What is the Z-disc? These are actual physically linked bands or walls of proteins. And if you need to know more about them, you can jump online. That's all my students need to know about the Z-disc for basic anatomy and physiology. But the Z-disc is a physical structure. It's a wall. It's basically a structure that things can grow off of, if you want to think about it like that. Let's do one more thing before we start to put in the different myofilaments. Let's put a dashed line right down the middle of this thing. Okay, right down the middle of the sarcomere. We're going to end up labeling this as the M line. M meaning middle, right? This is the M line. This is the middle of a sarcomere. And that's going to help us be able to do the rest of our drawing. Okay, Z-disc, and an M line down the middle. This is all part of our sarcomere. And now let's start adding some myofilaments to this. The first myofilament that I'm going to add to my drawing is not one that you normally see done first. Normally you learn about thick filaments and thin filaments first, and you can do that. Go look in a textbook if you want to learn about those first. But I'm going to do something that's going to potentially help you learn this system a little bit better. That's my hope. It's always my attempt. So what we have growing from the Z-disc, and there's, all here, all of these are going to have this particular myofilament on them. Same thing with the other side. We're going to see them over here as well. Okay, and they're going to extend towards the M line, but they are not going to go all the way to the M line. That's important. They're going to extend towards the M line, but they don't go all the way to the M line. And this particular myofilament, this protein, is called nebulin. Okay, this protein is going to be called nebulin, and we're going to put that in as our number 6, even though we're starting it with number 1, because why not? We do however we want to do. We can do what I want to do. Gosh. Nebulin. Nebulin is really interesting. Okay, we didn't find out about it until a little bit later in terms of our understanding of the anatomy of this structure. We didn't see it, we didn't find it using scientific methods for quite a bit of time after everything else. Hence nebulin, it was nebulous. How did everything work? We don't know, it was nebulous. So what is nebulin? Nebulin is a scaffold. It's a ladder. It's something that is basically a stiff protein that just kind of extends out towards the M line and doesn't really have a lot of function in and of itself, and I'm sure there's some scientists out there that'll yell at me for saying that. But the most important aspect of nebulin for you guys to know is that it's a scaffold. It's something that another protein can attach itself to. And what protein is that? It's actin. And so actin, through a process called polymerization, actin starts to wrap itself around nebulin. And you're going to have individual G-actins that end up producing this long strand of F-actin. And then you're going to get 2 of these F-actin. Okay, and they wrap around each other and they wrap around the nebulin underneath. 2 F-actin make 1 actin. Okay, 2 F-actin make 1 actin. And so I'm just going to draw these down here. I'm not going to draw them on every single diagram. You guys can do that at home. It'd be a waste of my time and your time to watch me do that, I swear. Okay, so I'm just going to end up wrapping this on this side and wrapping around on this side. So basically just circle around. Doesn't need to be perfect for the case of studying. Okay, you're going to look at diagrams online. They don't look exactly like mine, but that's fine. So this structure right here is actin. I'm going to put that down as my number. We'll go up here to number 1. This is actin. It's one of the major myofilaments that helps with contraction and relaxation. What else do we need to know about? Let's go ahead and draw in another major myofilament, which is called myosin. And I didn't draw these perfectly lined up. It's okay. It's okay. It doesn't have to be perfect. Okay, my myosin's going to be a little crooked, and that's not the end of the world, I promise. For the sake of studying, it's okay. Okay. So there is my myosin. Myosin is also known as thick filament, where actin is known as the thin filament. And the reason why is because myosin is thick. made up of multiple myosin tails, which are these big long strands of proteins that attach here on the end line. And then they go the other direction. And each one of these myosin tails attaches on the end line. And every one of those myosin tails is going to end up extending up and out. Okay? It's going to extend up and it's going to end in a little head that we call the myosin head. Again, we're just basically covering the anatomy of the sarcomere today. Each one of these is going to have a function as well. But the myosin heads, okay, this right here is a myosin head, whereas the rest of this is myosin tails. That didn't come out as well as I wanted it to. One second. Let's get rid of that and draw it up a little bit higher. Myosin tails. Most of the thick portion is just lots and lots of myosin tails that are wrapped around each other and extending out, reaching out towards the actin. We have all these myosin heads. And this will be the number 2 thing we write here is myosin. Myosin, remember, is made up of both myosin tails and myosin heads. Okay, let's go down here. You can draw all these on top of one another, and we're going to do that in the very end, but I want you still to be able to see each individual piece of the puzzle. So we're going to go down here, and we're going to make another actin that wraps itself around nebulin. It polymerizes to nebulin. By the way, nebulin becomes pretty important there because the length that nebulin is determines the length that actin is going to become, because actin is too flimsy to just polymerize and grow out on its own. It needs a stiff scaffold to be able to polymerize on. Now, wrapped around actin, like a couple of Twizzlers, wrapped around actin is another myofilament called Tropomyosin. Okay, so here in red, we're going to have Tropomyosin. Tropomyosin we'll have here as number 3. Tropomyosin. And Tropomyosin covers these really important binding sites found on each G-actin. Each G-actin has an attraction or a high affinity site for the myosin head, and as long as tropomyosin is covering that binding site, then the myosin head is not going to be able to interact with that G-actin. So tropomyosin covers that high affinity binding site for the myosin head. So how do we get that tropomyosin to release itself? How do we get it to move off of that myosin binding site? That's what the 4th of these myofilaments is going to do. The 4th myofilament we're going to talk about is actually a protein. We're just going to draw it in as kind of this ball shape here. We'll fill in that ball shape here in purple. Okay, this one's called Troponin. Students that have had me before, or students that are actually in my classroom, they know that I like to go Troponin, like Carlton from Fresh Prince of Bel-Air, right? So find something in everything that you do to help you remember that system. Troponin is going to be really important because that is our calcium binding site. That's where calcium is going to bind, and that's going to initiate muscle contraction. And how it does that is troponin is attached to the tropomyosin, and so when calcium binds to that troponin, it's going to cause a physical conformational change that moves the tropomyosin off of the myosin binding site, exposing that binding site to the myosin head, and myosin head can finally can finally attach to that actin underneath. So this right here is troponin. And that is the end of our thin filament. So what I'm going to do is, using pink, I'm going to actually say that actin, tropomyosin, and troponin all together are what we know as our thin filament. So those three things combined make our thin filament. Versus myosin is the only major, it's kind of a hard thing to say. Oh, why am I writing myosin? Because I'm in the middle of another thought process. That's why. Myosin really is what makes up the large majority of our thick filament. You're going to see that there's more to it, and if you read in your textbook and stuff, there is more to the thick filament because it's where the actin overlap is found. There's actually titin, which is going to be the last myofilament that is going to be bound. But titin is really thin, so it's not going to take up a lot of the thick filament portion. Myosin is the big thick portion. Okay. At this point, we've pretty much drawn in everything except for one major thing. Okay? And that's the one that I just mentioned, which is called titin. And titin, we are going to draw in using the color blue. Most of your textbooks are going to show titin bound to the Z-disc, and it looks like a spring that just comes from the Z-disc and then binds to the end of the thick filament. That's what most of your textbooks are going to show you, and most of your textbooks are wrong. Okay? Most of your textbooks are wrong. That's not how titin looks. You can think about its function. Let's draw it back in. You can think about how it works this way. Okay? It kind of acts like the shock. The shock's in your car. So it acts as a gigantic spring. So if you were to pull on that spring, well, what do we know about springs is they have elasticity. So if you pull on that spring, boom, it wants to return to its original position. It's going to recoil. And if you compress that spring, it's going to want to boom, return to its original position. So that's why drawing it like this is okay. And it really is. If your teacher allows it, go for it, right? And I will allow my students to do it too. But just for the sake of good old titin over here, the largest protein known to mankind, that's what I studied through graduate school. Titin is actually, there are two of them in between each actin. So there's actually a titin that starts here, and there's a coiled element, okay, and then that coiled element goes down, and it attaches to the myosin, but then titin actually continues all the way to the M line. And then there'd be another coil here, and then that one extends all the way to the M line, and then there'd be a coil over on this side, which extends all the way to the M line. You guys get the point. There are actually multiple coils. They're bound in multiple positions, and each part of titin is really important in determining the elasticity, boing, the recoil, right, and the stiffness of your muscle. And that's about all I want to talk about titin in this particular lecture format. So let's pretend that that little skip didn't just happen. And what we're going to do is we're going to finish off our drawing just by putting in our tropomyosin. So we're just kind of wrapping it around our actin, all of our actin. You only need one band of it to understand how it works, because that's what it's all about. And then we're going to attach our troponin. You don't need these gigantic troponin way over here. Just draw in little dots. That's enough to understand troponin is there. You can label them. And then you begin to understand the function better of each one of these things. Okay? So now when we look at this, we can say, hey, troponin is the calcium binding site. And when calcium binds to troponin, something's going to happen. Well, what's going to happen? Well, troponin is bound to the tropomyosin. So when troponin gets activated by calcium, it moves the tropomyosin off of the myosin binding site found on actin. And that's where those myosin heads are going to attach to, and then something else is going to happen. To find out what else is going to happen, go watch my YouTube video or any YouTube video on the cross-bridge cycling and how the sliding filament works. sliding filament theory. So that's the end of my little presentation on the sarcomere. It's not everything because we can't possibly have a video on everything. So get what you can out of this, be able to draw, fill it in, put in more thick filaments, put in more thick filaments, label things like the I band and the I band. You're going to have to go in and start to learn about the H band as well. But I'm not going to spend this entire video putting that all together. Okay, this is just a video of how can I quickly draw a sarcomere. So if you need to be able to draw a sarcomere even faster for an exam, check this out. Okay, now we got Z-disc, Z-disc, thin filament, thin filament, thin filament, thin filament, a thick filament in the middle of each one of these. You don't need to draw them in. Okay, then take and maybe grab a different colored marker, wrap it around, that's your Tropomyosin, Tropomyosin. Then you drop your Troponin in there with little dots. Okay, you don't have to be perfect. This is for an exam for you to be able to understand a question, like a multiple choice question or something along those lines. You just need to have a diagram that helps you remember where everything is. Okay, you could put in your extensions, your cross bridge heads. If you need to talk about them, you just draw them in. Make sure they're pointing towards the Z-disc. Again, it doesn't need to be pretty, it doesn't need to be perfect. It just needs to be a nice, fast diagram that helps us understand the system a little better. And that's where the whole, just a single Titan's coil on either side, that's where it comes into play. It just makes it faster and easier to draw, so now we throw in Titan. And that's a nice fast way to be able to draw out a sarcomere, to be able to understand what the different relevant pieces of the sarcomere are. I didn't even put in nebulin, but you can understand that actin polymerizes around that nebulin filament. Hopefully this is helpful in your understanding of how to draw the sarcomere. In order to understand how its function works, you'll need to look more at other videos. Again, like cross-bridge cycling video. All right, thank you for spending some time with me. And remember, humankind, be both. See you in the next video.